Hello, Namaskar, Kambagani people, and welcome back to our channel, Professor Tiwari, and my name is Amrita. So yesterday we discussed, in the last section we discussed about the endomembranic structures, that is the endoplasmic reticulum we have covered the Golgi apparatus or Golgi bodies we can call it, and we have covered the lysosomes. The last part that has been there in the endomembranic structures was the vacuoles that we need to cover. So let's go for the topic further. Okay, so today we are going to discuss the vacuoles first. Okay, vacuoles. Now what these vacuoles are? See, vacuoles are basically, these are the membrane bound structures. If I call them, they are membrane bound spaces, you can call them. Okay, they are membrane bound spaces. Now if you just look at the structure of the this vacuole, it is like this. Okay, and in between that it contains a central vacuole. Alright, so if I have to just name it, I'll call it, so this is your vacuole, this hole is your vacuole, the layer, this membrane outside the vacuole which is covering the vacuole is being called as tonoplast. Okay, inside this whole area, this lumen of the vacuole or the space of the vacuole, inner space of the vacuole is filled with cell sap or water, you can say, and this one, this small one, is called as central vacuole. Okay, now if we just look at, so what does this vacuole contain? So this vacuole contains cell sap, water, some excretory material. Excretory material is actually that material which is the waste material generated by the cell due to the cellular respiration which has been there, and there will be other non-useful material you will find inside this cell sap, okay, which is present in the lumen of it. Now if you talk about the lumen, the pH of the lumen is acidic, and it is approximately same as that of the lysosomes that we have studied, which is the 5. Okay, so it is acidic, the inside of the lumen the pH is acidic and it is around 5 in nature. Okay, if you talk about the cell, if you talk about the vacuole of the plant cell, this is like the animal cell. If I talk about, if you talk about the vacuole of the plant cell, so this is your plant cell. Okay, and this is your vacuole. Alright, this is your vacuole. So basically what happens in plant cells, if I talk about, this is for the plant cell and this is for the animal cell. Okay, in both of these the structure is same. You will have the membrane will be, the membrane of the vacuole will be of the tonoplast only. It is filled with cell sap, excretory material or some other things, and it has a central vacuole too. But the major difference between the vacuole between the animal cell and the plant cell is that the plant cell actually occupies the 90% part or 90% volume of the whole cell, plant cell. So occupies, when you talk about plant cells, when we say about plant cell, so in plant cell what happens, occupies 90% volume of cell. Okay, so it occupies 90% part, plus it is permanent in nature, whereas in the animal cell the vacuoles are temporary in nature. They will come up when they have to do the cleaning procedure, and then they will dissolve away, okay, or they will move out in any of the cases. All right, now moving ahead into this one is that the role of the vacuole in, what is the role of the vacuole that we have studied, that it removes excretory material, this and that. But what about a tonoplast? See, a tonoplast plays a very major role over here. What it does is that this tonoplast, the membrane, this tonoplast, especially in plants, it actually facilitates active transport. It facilitates active transport. When we say active transport itself suggests that it will use ATP, it will be with the use of ATP only, and it will always be against the concentration gradient. Okay, which means what happens is, outside the cell in the cytoplasm there will be materials, okay, which has to go inside the cell. Okay, so now what happens is, the tonoplast will start proceeding, or will start doing the, it will start doing the transportation. Now this transportation will be with the help of ATP, that is why it is being called as your active transport, and it goes against the concentration gradient. When I say concentration gradient, which means that the amount which is being there present outside is always lesser than present in the inside. Okay, which means the inside, the plant has more water, it has more cell sap, it has more excretory materials, so which means it more of like, still there are some water, extra water or extra excretory material has been there outside the cell in the cytoplasm, so it has to go inside. So the chloroplast will run the active transport, and it will go, and all those materials will start going inside with the help of active transport, or you can say with the help of ATP, and always against the concentration gradient. When I say against the concentration gradient, which means from higher concentration to lower concentration. Usually it is lower concentration, higher concentration always, but here it is higher concentration to lower concentration, which means there will be higher outside, so they will start moving inside. Okay, now moving ahead into this one. Other than these vacuoles in the animal and the plant cell, there are two other type of vacuoles which are present. One is in the amoeba. Okay, now this amoeba actually contains contractile vacuole. Contractile vacuole. Structure is same everywhere. This contractile vacuole, what is the role of contractile vacuole? Excretion. Okay, why contractile vacuole is important? Whenever the amoeba engulfs the food and it digests, because it is a single cellular organism, so it proceeds with the digestion system, with the digestion, and once the digestion is been there, the waste material gets collected in the vacuole, and that vacuole has been called as contractile vacuole, and it provides the function of excretion, which means it will fuse with the membrane of the amoeba, and it will throw the waste material out. Now another type of vacuole which is present in the cell, which is your protestis. Now this protestis actually contain food vacuole. Now what this food vacuole does, it actually engulfs food. Okay, it engulfs food, which means when the food has been there, because again it's a single cellular organism, so it will engulf the food from the environment and will take in, and the digestion process starts. Okay, so there are these two more type of vacuoles are been there, which you will find in amoeba and in protestis. Okay, now moving, so this was the last part that we have studied in the endomembrane structures that we have started in the previous lecture. Now we'll move ahead with the another type of a structure, which is not the endomembrane structure, but it is an important cell organelle which we need to study. Okay, and without that organelle, the whole cell cannot work, and that organelle is being called as your ribosomes. Sorry, not ribosomes, mitochondria. We are studying mitochondria, which are the most important one. Okay, now we'll study about the mitochondria, the third one. Okay, before we study mitochondria, let us draw one diagram for the mitochondria. Now I'll just draw the outer one. So mitochondria is basically, if you just look at the structure of mitochondria, this is a mitochondria. Very interesting thing. So if you just look at this one, this mitochondria, this is your outer membrane, this one is your inner membrane. Okay, inside the mitochondria, you will find the DNA. Okay. Other than that, you will also find ribosomes. Other than that, you will also find one very important thing which is present near the membrane. Okay. Alright, and if you just magnify the structure of this one, I just use this color, I just magnify the structure of this one. I am magnifying the structure of this one over here. It looks like this. Okay, this part is being called as F0, this part is called as F1. Okay, they are being called as F0 and F1 portion. Okay, and this is your intermembrane compartment. Okay, now moving ahead into mitochondria. Mitochondria is actually a plural word, which means when lot many mitochondria are there, large in number, we call it mitochondria. If it is a single, if you see a single mitochondria, you call it mitochondrion. Okay, you call it mitochondrion. Okay, now what it is? Double layered. One is this one, one is this one. Mitochondria is the only organelle present in the cell right now which is a double layered structure. Okay, it's a double layered structure. You will find the inner membrane and outer membrane. Okay, if you just look at the number of mitochondria, if you just look at the number of mitochondria, then in that case the number of mitochondria actually varies. Number of mitochondria varies. How it varies? According to physiological activity of the cell, which means which activities the cell is performing, so which is important. Other than that, it is also on the shape of the cell, what is the shape of the cell and what is the size of the cell. So it depends on that. Okay, now if you just look at the size of the mitochondria, then mitochondria usually has a diameter of 0.2 to 1.0 micrometer. This is the size of your mitochondria, diameter I would say. Okay, this is the diameter of your mitochondria. And if you just look at the length of the mitochondria, it ranges from 1.0 to 4.1 micrometer in length. This is the size of the length of the mitochondria. Diameter is this one and this is your length of the mitochondria. Okay, now talking about this double layered membrane of the mitochondria, then this double layered membrane is actually divides the lumen of the mitochondria into two parts. Okay, the lumen gets divided into two parts, the inner component and the outer component. Okay, now what this does? Membrane actually divides lumen into two parts. Just look at it. Inner compartment, which has been there, this one, and your outer membrane. If you look at the outer membrane, you will see that outer membrane is actually continuous, which means there are no foldings being there. Okay, like the structure which has been there with the inner membrane. So this outer membrane is actually continuous. Okay, whereas if you just look at the inner membrane, this inner membrane is actually has foldings. If you just look at it, has foldings. These foldings are actually being towards the matrix or matrix. It is towards the matrix. This inner portion is being called as matrix of the mitochondria. So this foldings are always towards the matrix, which means inside the matrix, like this. If this is the lumen, then it will be like this. So this is towards the lumen. Okay, it will be towards the matrix, and they are actually being called as cristae if they are in large numbers like over here, and if you just need to pronounce it in single way, then you call it crista. Okay, all right. So single, if it is single, for this single one you will call it crista. For this one you will call it cristae. Okay, now moving ahead over here is that if you just look at this one, Okay, now these things, these are your F0, F1 particles are been there. You will always find them in the, they will find them in the matrix only. Okay, now very important thing is that both of these compartments, the outer compartment as well as the inner compartment, they have both membranes. Both membranes, or you can say both compartments, have separate enzymes for them. This will have its own enzyme, this will have its own enzymes, okay, for the cellular activity of the mitochondria. Now moving ahead into this one is that, now as we have seen that mitochondria has its own enzymes, and which means it is performing its own functions. Now an important thing over here is that this mitochondria is actually, I said that we are going to study a very important topic, that is mitochondria. Why this mitochondria is so important? Because the mitochondria is actually the site of aerobic respiration. What is aerobic respiration? Where we use oxygen. Oxygen is being used, aerobic respiration. What it does, what it produces? It actually produces ATP, which is the energy currency. Okay, and because of this reason, mitochondria is being called as powerhouse of the cell. Okay, this is being called as powerhouse of the cell because of this reason only. Talking about, as I told you that it contains the enzymes. Now if you just look at the inner membrane, so inner membrane is actually impermeable. When I say impermeable, which means it will not allow, it will not permit anything, especially solutes, impermeable to solutes. It is impermeable to solutes, but it is rich in phospholipids. It is very rich in phospholipids. One of the such of the phospholipid is cardiolipin. Okay, cardiolipin is one such phospholipid which is present in the inner membrane. It is very rich into that. Other than that, inner membrane, it also has a very important enzyme which we will call as ATP synthase or F0, F1. When I say F0, which means F0, F1 ATPase. Okay. Other than that, a very last thing which has been there over here is that mitochondria is the only other organelle present in the cell which has its own DNA. So it contains a single circular DNA, okay, which is present in the matrix only. It contains few RNA molecules you will find. Okay. Other than that, you will find the ribosomes. Okay, that would be the 70S1. So basically mitochondria is called a powerhouse of the cell because it generates the ATP, which is used as the energy currency of the cell, which is important for all the other functions that the cells or the body has to perform. Because of this reason only, mitochondria has been termed as the powerhouse of the cell. Other than the mitochondria, there are some other organelles which also produce ATP, or some other reactions are been there which produces ATP, but they are not being produced in a large quantity. Mitochondria is the main one. Other than that, mitochondria, the inner membrane of the mitochondria, the inner portion of the mitochondria is actually rich in the phospholipid, and one of the phospholipid is the cardiolipin. It is impermeable to the solutes, which none of the solutes can enter into it. Okay, none can leave to that. Inner membrane is also they also have an enzyme complex which is being called as F0, F1 ATPase or ATP synthase, which has been there. Mitochondria also is the only organelle which has its own DNA, which is a single circular DNA it has. It has its own ribosomes which are being present over there, and it also has few RNA molecules which has been there, and it produces its own protein. Okay, that is why the mitochondria can divide also. Okay, so that is all for today. We have discussed the mitochondria, we have completed the endomembrane structure. In the next class we will move ahead in the next video. If you have any doubts, you can drop them in the comment box below. Please like, share and subscribe our channel, and don't forget to hit the bell icon for more such updates. Bye. Thank you.